I'm going to talk to you today more about how my life sort of as a working scientist as opposed to as a teacher, as an instructor, has developed and makes use of integration with social media, because it's been a big part of things that I've been doing for several years now, and I just kind of want to present our side of the story. I think a lot of it's pretty ad hoc, right? Like I use things to try and make things happen from time to time, or just to get the word out. So I'm not an expert in the use of social media as a scientist, but I am a scientist who uses it as much as possible. So hopefully there will be some take-home messages that you guys can benefit from that that come out of just some reflection that I've done on how I've been using it over the years. I am on Twitter. That's going to become obvious how important that is, I hope, by the end of this. That's our lab website. And then the website at the bottom is going to be something I talk about a little bit more as well. All this is, I guess, being streamed live, so hello world. It's going to be recorded, so you don't have to freak out if you can't see something right now. I'll also make these slides available as a PowerPoint at the end too, so you have everything in it. So just relax, enjoy. I think before I get started on talking to you about social media integration, I want to spend a few minutes though just talking about the science, some of the science that we do in the lab, so you have some context, right? Like, it's one thing for someone to be integrating social media when they're working on science communication research or education research. It's a completely different thing if they're a physicist, an astronomer, an astronaut, or maybe like us, microbiologists. So I'm going to start there. I'm going to proselytize a little bit about our microbiology research just to give you an introduction, and then come back around to talking about how we make use of social media, blogging, and internet integration just in general to kind of get ourselves out there, but also to bring other people to us and to the information that we're creating, and then also to get people to interact with each other, not necessarily through the context of the work that I'm doing. So I'm a marine microbiologist. I study bacteria in the ocean, bacteria in Archaea, if you're a stickler. Prokaryotes in general, these are small organisms, usually around a micron or two in size. And what I have up here is a picture from a microscope image. Is there a way to kill the lights just right here?
You did the, yeah.
Well, kill all the lights. Okay.
You
that's
can
fine.
leave that last one on.
Yeah, that's okay. That's good too.
Yeah.
So what you have here is a microscope image of seawater that's been stained with a nucleic acid specific dye and then just imaged with fluorescence microscopy. And most people, when they see this, think this looks like the night sky, looks like stars, and that's not far from the truth in terms of numerical abundance. One of the reasons why I like to put this slide up is because when we're talking about organisms that live in the ocean just naturally occurring, we're not talking about pathogens, we're not talking about things that will harm you, we're just talking about organisms that are naturally occurring. They typically in the 10 to the 5th to 10 to the 7th cells per milliliter. So in an eyedropper full, roughly, you have anywhere from 10 to the 5th to 10 to the 7th cells. 10 million cells, maybe a little bit less, 100,000 cells. But when you add up all the water in all the oceans, that gets you a really, really big number. 10 to the 30 is an estimate that gets tossed around a lot. And so we're talking about very small creatures, but we're talking about them in massive quantities. And so their impact on the way that global cycles on the planet work is huge. Carbon cycling, nitrogen cycling, sulfur cycling. And a lot of the organisms that are here, we don't really understand all that well. We can zoom in on an image like this, use a little bit more technology. This is a scanning electron micrograph of seawater that's been filtered onto a 0.2 micron filter. So if you just go out in the ocean, you run seawater through a very, very small filter. These are the filter holes right here, and then fix and image your filter. You can see a massive variety of different forms of organisms that are present in seawater. Again, keep in mind, this is 0.2 microns, all right? And just for context, 1 micron is about 1,000th the width of a bicycle spoke. All right, bicycle spoke, pretty small. Slice it into 1,000 pieces lengthwise, that's 1 micron. This is 2/10 of that, 20% the size of that, and those are the sizes of the organisms we're dealing with. And so this, I love this image because it frames our problem really nicely. These things are hugely abundant. Soil, air, on your body, in your body, out in the water, they're all over the place. But they're very small, and so trying to figure out what they do and how they do it and why they are where they are becomes a pretty big challenge, all right? And I sort of use this image to frame a question that we're really trying to understand, which is why is that microbe there? You pick one. Why is it there? And maybe a little more focused, why is it there as opposed to some other organism? In other words, what is unique about its properties? Does it have special functions? Why does it show up at some times of the year and not others? How do we figure these kinds of things out for things that are very, very small, but nevertheless important? And that's really what frames the research in my lab, is trying to do this for certain groups of organisms that we find in certain situations. And really what we're focused on right now is the coast of Louisiana, the northern Gulf of Mexico, and the waters that are actually pretty close and important to us here in Louisiana. Now, if you want to go study a microorganism, you probably would go about it with the same ideas that you would have for studying a tree or a deer or anything else. You want to go watch it, see what it does, how does it interact with other things around it, what does it eat, how does it act when it's scared or when it gets cold or when it gets warm, does it breed, if so, how often? These are all kinds of things that you would want to do. We want to do the same things with microorganisms, but again, they're very, very tiny, and we can't see them with the naked eye, and they're surrounded by lots of other microorganisms that are different from them. Some of them are similar, some of them are really, really different. So how do we go out and observe them? How do we get these organisms to a place where we can interact with them? One of the things that we try to do is bring them back into the laboratory, just like you would some other organism. If you wanted to study how mice function, you want to study what's inside of a mouse, you would want to bring it back into the lab and you would do experiments on them, or you would do dissections on them, or things like this. Same thing with trees or leaves or anything else. So that is not as trivial as it sounds for various microorganisms. If you've taken microbiology laboratory, you're used to streaking out E. coli on a plate or something like that, and you get things to grow on plates. But most microorganisms in the ocean actually have a problem that we describe as the Great Plate Count Anomaly, which is essentially that if you try to take water from the ocean and put it onto a plate of normal media, you get 0.1 to 1% of the total cells that you would see under the microscope. So we're doing traditionally do a really poor job of bringing those organisms into culture to play with. Now let's just back up for a second. What are we talking about with microbes? This is a tree of life that was created in 1997 based on the 16S ribosomal RNA gene. It basically, or the small subunit ribosomal RNA gene, it's 18S for the eukaryotes. It basically gives us these 3 large branches of the tree of life. These organisms, well, you are here, Homo, right next to it. Zea, which, does anybody know what Zea, the genus Zea is? It's corn. Right, so you're here. All of these organisms along these early diverging lineages are microorganisms. They're eukaryotic microorganisms, but for the purposes of our laboratory research, we're really focused on these 2 massive branches here, or what we call prokaryotes, bacteria and archaea. Things have changed in the last few years. In the bacteria and archaea, we have dozens and dozens of phyla that have been identified really just by sequence information from the environment. So we go out in the environment, we PCR the 16S gene from DNA that we extract from the environment, and we realize that there are a lot of organisms out there, but only the triangles that are colored white have cultured representatives. In other words, those are the only places where people have been able to get the organism, bring it back into the lab the way you might hope to. All these other branches that are black or gray, we actually don't have a cultured representative. We don't know what those organisms do. And so we have this massive diversity of creatures that are out there. Each one of these is a branch that represents as much diversity as all of animals, or something potentially even greater sometimes. And so we know there's a ton of diversity, a ton of interesting organisms out there, and most of them we don't have in culture. We don't have them in the lab to play with. And so one of the things that we do really in my lab is sort of a two-pronged attack where we try to use cultivation and cultivation-independent methods to understand these organisms. All right, so some of our efforts go to really trying to overcome the great plate count anomaly and get these organisms into liquid culture. Some of our work tries to understand these organisms by just extracting the DNA from them. Okay, we can't grow them in the lab, but we can get pieces of their genomes and we can learn how to understand them that way. And that's sort of the way that we go about it here. We also combine all this information with detailed geochemical measurements. So we want to know about nutrients. We want to know about how much daylight there was that day, how salty the water was. And we do this from a seasonal perspective as we move forward, sort of taking samples all throughout the year over the course of several years to try and understand how these organisms are interacting with their chemical environment. So we have developed, well, we have not developed, we have extended a method that was developed by others for overcoming the great plate count anomaly, and that's by using a couple really interesting methods. One is dilution to extinction culturing, all right? And essentially what this means is you take seawater from a natural sample and you serially dilute that seawater until you've got basically one microorganism on average for any given culture vessel that you're using. The other thing that you can do is you could take natural seawater and instead of using LB or the plate-based methods that we had before, you filter and autoclave that natural seawater and then you add your organism to a culture flask and you let it grow in the type of thing it was already living in to begin with. Okay? Now this works for a couple of reasons, and it's all based on an idea that most of the organisms that are prevalent and abundant in the oceans don't like growing around other organisms very well. They're fine growing around other organisms, but they grow very slowly. And so if you put them in media with other organisms, those other organisms will outgrow them and they will get lost in the weeds, so to speak. The other thing is that we don't really, believe it or not, understand all the components of seawater very well. We know the big things like how much salt there is and how much phosphate and iron and stuff like that. But the dissolved carbon components, nitrogen components can be very, very complicated. They can be very numerous and diverse. And really, we have a poor understanding of what it is that these organisms require in their natural media. So using natural seawater is not a bad way to go. This is our version of the workflow. I'm only going to have one more slide after this on our science. So this is just to kind of introduce you to the way that we're working on all these problems. We start by collecting water. We have a flow cytometer that we use to count the number of cells that are in there, which helps us do that dilution process and figure out how many microliters of liquid we need to dilute these cells with to get us down to 1 cell per well or 3 cells per well. We have a medium distribution process into custom 96 well plates that are 2 mils deep. We sit them in there with that natural media. We let them sit for a couple weeks. We go back and count them. And then we will get some of these organisms will grow. And we'll have what we call positive wells, which is where we've got cells to grow. And then we do a whole series of things with them. We go out and transfer them into larger flasks to try and get more biomass so we can start doing experiments with them. We extract their DNA so that we can identify them and figure out who they are and what they do. We preserve some of them. This is like backing up your computer, backing up your hard drive. You know, we get many, many copies of the same organism by growing it up. We put some of those away, put them in the freezer in case we lose the culture, something bad happens, it dies, or just as time goes on, it evolves and mutates and changes into something that's a little different than what we started with. So we've got a hard drive, we've got an identification system, and we've got our experiments going. We do a couple other things. One is that we filter the water directly, extract the DNA from all the cells on those little filters, and then we go back and enumerate how many of what kind of cells were in that original mixture so we can decide and figure out which organisms are important. Are some of the ones in these flasks the ones that are the most dominant in the system to begin with? Hopefully. Are they organisms that we know are important because of their interactions with some of these different nutrients in other systems? Maybe that's a way to help guide us which of these organisms we want to study further. And then, like I said, we've got all kinds of additional chemical analyses that we could perform on these. Okay, so this is kind of a complicated workflow. Some of these concepts are a little esoteric. They're not relevant to everybody. But there's something really cool about discovering new organisms that I think almost anyone can relate to. And so in there lies the ability for us to communicate with other people, and I think allows us some you know, a foothold for captivating an audience, all right? I think we have some material that other people might find enjoyable, even if they're not microbiologists. The other thing that's really cool about what we do is this part, the collection part, because we get to go out and do field work. And this looks really neat most of the time. We're collecting at 7 sites around southern Louisiana for this culturing process. We're also collecting in deeper water all along the shelf area of the Gulf of Mexico, because we study the dead zone, we study the microorganisms that are present in low oxygen. We study a station out here off of an oil platform that we go to every few months to sample, and we do that with scuba diving. And so as a result, we have images of things happening around the coast of Louisiana that are actually kind of cool, even if you're not a microbiologist. We've got the fort out at Lake Bourne. There's dolphins that we run into in this, jumping into the sunset. This is one of my graduate students who's motoring our little research vessel, my 14-foot aluminum John boat with the 10 horsepower engine. That's our illustrious research vehicle. This is a beautiful image of the Calcasieu jetties over in southwestern Louisiana. And so we're interacting with a lot of really cool environments. The other thing that we have an opportunity to do, which is useful for social media integration, is interact with people that are interested in some other issues. This whole part of the coastline is under massive sort of, I guess attack is probably a little too aggressive of a word, but it's certainly under significant human influence. We have oil spills around here. We have hurricanes that run into things. That's not human influence, obviously, but that's natural influences. We also have a ton of land loss that's occurring. The figure right now is a football field every hour of receding landlines, receding coastline, or just general marshes, land everywhere. That's because of a whole series of causes, most notably the diversion of the Mississippi River so that it's not continually replenishing all this. But there's a lot of activity, human activity, like cutting through all these areas to make waterways and do oil exploration that's causing problems as well. And we're not replenishing that land with our own efforts quickly enough to stave that off. So this is a changing landscape. It's a changing landscape due to a lot of human and natural influences, as well as a beautiful landscape. We've got some captivating imagery. And so all of this kind of plays into what we do with our social media integration, which is to not just talk about our science, but also talk about where we are, what we're doing, and how that may, and try to provide some context that's relevant to a larger audience, a much larger audience even than just microbiologists. Okay, so what I want to do now is actually kind of take you to some of the efforts that we have in my lab and some things that I've done in the past. So we're going to switch gears a little bit here and just get on the internet and mess around. My approach with social media has been founded mostly in Twitter. And this is mostly because there's a large microbiology community on Twitter. So already starting at the beginning, we've got a really nice sort of community of people, researchers that are interested in what we do on Twitter. And they're all having, not all of them, but a lot of them are having conversations. And it's really easy to find a microbiologist on Twitter, go out and ask them a question. For example, I saw somebody tweeted an idea the other day about a conference they were at and about some ideas they had about evolution. And there's a man on Twitter I know who's a National Academy member. He's very, very famous, and he's the foremost expert on the way that microbial evolution works in long-term evolution experiments. And so I just sent him the link from the talk that was being live tweeted on Twitter and asked him if he saw the same types of observations that this person was postulating happened in their environment. And all of a sudden we were in a conversation about theory versus actual observations and how those things may change over time. It was really neat. We use Twitter integration in my prokaryotic diversity class, as Colleen can attest to. And we had a few examples where students had questions about certain specific things that they'd read or seen or been interested in. and we're able to just tweet at a professor or somebody who actually did the study and get answers back very rapidly, like within minutes. So it's a really cool way to integrate with people, to ask questions, to talk to folks too. But one thing I want to point out here is you'll see on my page, right here is just all journal titles, okay? As a scientist, one of the things that I'm tasked with is making sure that our work is contextualized with the work of everybody else. That means knowing what everybody else is writing about and working on. If you've ever done journal research for a paper, you already know what that's like. You've got to go find primary literature sources. You have to understand what people have written about something. This can take some time. I monitor over 100 table of contents from different journals every single day. Otherwise it gets pretty exhausting. So one day I was doing this and I decided, well shoot, I might as well tweet the papers that I find because they might be valuable to somebody else. And that's become something that's been really successful for me personally in terms of getting attention, because a lot of people check the feed to look for journal titles. That has in turn translated itself into a really nice way for us to get information out about things that we're doing in the lab. Because the more followers you have, obviously, the larger that broadcast message is, not just in terms of the people who read your tweets, but the people who retweet your tweets and the cascading effects from that. So the take-home message from this is that there's sort of some utility to doing something that's useful to other people as well as just yourself. And that's one of the things that I'd like you or one of the lessons I think I've taken away from social media integration in terms of being successful is that if you want to create a brand, you want to promote a brand, you want to move forward with things and get more and more attention and get the word out, it's useful if you're doing things that aren't just good for you, but they have some utility, something that's beneficial to the wider community. In other words, can you be a resource for other people rather than just an information spewer or someone who's just talking about what you like or things like this. So there's a few ways that I'm going to come back to that point later on. So Twitter's been good. I live tweet conferences. I think that's a really neat way to take notes. It's also a great way for people who aren't at the conference to participate. It's also another way for people to find out about you and for you to become a valuable person of the community. If you're at a conference or you're at a talk or even just a noon seminar, if you're broadcasting a lot and a few people are reading that because they're sitting in their office and they didn't get to go, that can get you more attention, but it can also be a really useful resource for the community as well. So that's another example of how that works. My students are on Twitter as well, and this is an opportunity that they have had to interact with people, like I said, from outside the community, but when they are working or blogging or doing things on our website, they have the ability to sort of transmit or advertise that information, and I think it's an amazing resource. I can't go on enough about joining a community of scientists on Twitter in terms of the opportunities it can provide you for giving a talk somewhere, meeting people at conferences, maybe getting a job interview. It's a great way to kind of buffer all that. Now, you will also notice that if you look through the photographs and the media that I have on here, there's no pictures of me in a bikini with a cocktail. I don't talk about my personal life. So I really have restricted Twitter to a very professional account. It's not something that I use like I would use Facebook. And that's something that I think when people are first starting, they may have to integrate, maybe start a new account, maybe do something, because as you gain followers and as you gain more and more presence, you have to be cognizant of the fact that there are suddenly a lot more eyes on you. And maybe most of those people aren't going to be people you don't really know what their intentions are. It's something that you want to try and be conservative about, right? And I think for students starting Twitter to integrate with science or professional situations, that's a warning you want to keep in the back of your mind. You can always delete things and go back and delete them and stuff, but that's deleting them from your view right now, and it's really hard to delete anything permanently off the internet. So it's just a word of caution as you start to think about ways to integrate with people via media like Twitter or Facebook or anything else. I don't use Facebook as a scientific medium. That's strictly for my personal life, and so I have 50 50 friends on Twitter or Facebook or something. It's really pathetic from from a social media perspective. But those are like only people that came to my wedding kind of people, right? It's like it's not even like people I used to know back when I was in high school. Okay, so that's sort of my perspectives on Twitter and that. But we do a lot of other things on the internet as well. I want to give you some examples because these were things here, blogging in the ancient past 3 years ago, these were the things that I was doing prior to getting my job as a faculty member here. This is while I was working as a postdoctoral researcher. So I had my PhD, but I had not yet gotten a faculty position. This is also, this part is going to be relevant to people who are graduate or undergraduate students, graduate students. In other words, basically getting the word out about yourself and your interests while you're still being employed by somebody else, while you're still working, doing someone else's tasks. You could be doing this as a technician, you could be doing it right now as an undergraduate. There are ways to sort of start generating attention and generating interest in yourself and in your work that I think can be super valuable. And from my perspective as a faculty member who's interested in hiring somebody, right, as an undergrad in my lab or as a grad student or a postdoc, these kinds of activities speak volumes about a person that you cannot get across on a CV. You may not even be able to get out of somebody in a 2-hour-long interview process. If you get to see how someone writes, how they think, what they're passionate about, by what they've produced out there in the world, just of their own volition, without getting paid for it, on their own time, it tells you a lot more about that individual than you can learn in any short amount of time with them or even with other documents, okay? So this is not just something about like advertising yourself. Really, it's just about being yourself, and in turn, that allows other people to be able to understand you a lot better from a hiring perspective, okay? And so I want to show you, this is a website, this is dead. You can't get to this link anymore because it's private. But this was what I was doing as a postdoc when I was working. And what is here is small summaries of the papers that we were writing. This is maybe more relevant to a postdoc or a graduate student than an undergrad, but even as an undergraduate, if you're working in a lab and you're producing some work, you create a poster, or even if you're reading a paper that you really like, you can talk about it and create small summaries, put your opinions up there. I have a lot of links here that I was using to try and make the lab a little bit more useful to people. The Twitter feed, you can integrate that right into there. So if someone gets a hold of your webpage but they didn't know you were on Twitter, they can see some of the things that you're doing. This is there's links in here, images that came forward from this. You pick like a single image that you think summarizes the work really clearly, maybe put a caption on it and blog about it a little bit. And so this was basically what I was doing. It's not an extensive blog. It's just summaries of those various papers. I believe there were, here we go, a list of all the publications linked so people can get to them. Education and outreach. This is extremely important the further along you get in your scientific career, because the world wants to know that they're not just spending money for you to go off and do your hobby and the rest of the world doesn't benefit at all. You need to make sure that everyone knows how the rest of the world is going to benefit from it. You need to go out there and explain it to people. You need to go out there and teach, volunteer, take that information, get it across to them. This is an important part of being a scientist, is communicating your findings. I don't have to say that too much in here because y'all are all about science communication already, but you believe it or not, it's kind of teeth pulling with some people to get them to interact with the lay public. So what I put in here was everything that I was doing that wasn't a publication, that wasn't something that I could summarize in a blog post really easily. I think there are even, just some links. But what I have here then, though, are blogging entries too, because what I did as a postdoc, and I want to take you over to this site, was I volunteered to blog for a ocean conservation, where did I put this? Here we go. An ocean conservation group called One World, One Ocean. And they have a blog there where they do all kinds of different things. But what I was able to do here was talk about things like going on research cruises, Noah's largest research vessel. So I was on a week-long cruise in the Atlantic Ocean, and I took a bunch of pictures, and I talked about the work that various scientists on this cruise were doing, and got it onto the site, basically kind of like as a reporter. This was a really valuable opportunity to me because it taught me how to write really, really non-technically. Luckily, I had actually an editor that I reported to, and I would send the stuff in, they'd be like, nobody knows what that word means. You're not clearly communicating this. This doesn't have enough pizzazz. This is too technical. This sounds interesting, but you just glossed over it, right? Types of writing tips that you can always benefit from. And in science, it's usually the opposite. This is too simple. You need to put more detail in, or things like that. But constantly adapting your writing for different audiences is a really good exercise. And so the more opportunities you can create to do that, the better. Also with this, and another thing about Twitter, is that they limited me on every one of these blog posts to like less than, this is probably the longest one I ever did, but they would limit me to like 500 words or less. So it was like, get your message out there, be concise, make sure everybody knows what you're trying to do. Don't go on and be mouthy about it. Same thing with Twitter. I love being able to have small snippets of interactions. Live tweeting conferences is exactly like this. What can you say in 140 characters or less, now that you have to include hashtags for all these things, that gets across the point of what you just heard? Boom. Can you do that or not? It's a great brain exercise. Colleen, did you think it was useful learning how to do that in the class?
Oh yeah, definitely. Because I mean, as like scientists, like you want to talk about everything, but you really only have 30 seconds to grasp someone's attention. So like being able to take those scientific papers that like you made us read and then take the major things from it in under 140 characters, it's like a really valuable exercise.
Good. Okay. So I've heard some other feedback like that before. I think that the time element is something that I don't even appreciate enough. It's sort of like nobody even wants to read it, even if you did write all that stuff. They want the quick information so they can grasp it rapidly. Practicing being concise is valuable no matter what kind of writing you're doing. So I think that that's another thing that was great about doing this blogging exercise. Okay, so let's talk about what we're doing now. Since joining the department, I've set up my own lab website, like one does. And we have a whole bunch of static pages, like the research that we're doing, the publications, the people in the lab. We're going to slowly have the collection of all the microorganisms that we're culturing. We have over 40 at the moment, but I've been lazy and I haven't gotten this table up there yet. But we're going to have that link pretty soon. Then there's some other things, and these are places where we really add utility to the site in terms of providing people with a reason to come to the website that maybe extends beyond just the things that we think are cool. The front page is a blog, and what you're seeing here is a post by one of my other graduate students, Mike Hinson, who went on a cruise, and he live tweeted the cruise, and he posted some of his video. This is just this is giving you a sense of what it's like to work on the ship at night. That thing did that every 20, 30 seconds, and they'd be there for 24 hours. That can get, it's quick. It's a short video. You don't have to watch it for a while. It's short enough that anybody would click on it if they saw down below 7 seconds. That's a really useful thing to do. I didn't tell him to do that. He just came up with that. I think it's a great way to get the image across, the sort of the point of what he was talking about. Like, this is annoying. Great photos, and that was it. He didn't go on and on. Sometimes, though, there's a lot more writing that goes together with the pictures, but it's a great way for people to get a feel for what it's like to go out and do research. When we publish our research, we never get to put all this stuff in there. You can't put pictures of yourself pipetting on a ship in a scientific publication, but that's what people want to know about when you talk to them about your research. They want to know, well, what's it actually like to go work for this person, or what do you guys actually do? You can read the paper and not really know what it's like to do what we do. This is what we do. Look at this. My graduate student's been in the lab for just over a year, and he's already seen a water spout. I don't know if you can see it, but that's a tornado out in the ocean. I've never seen one of those. I was so jealous when he posted that. You know, pictures of people working on the ship, the way it looks to be, you know, tied off to a platform. Some of our sampling protocol or sampling information. And sometimes you get a little bit more wordy with these, and sometimes it's just pictures, and you just blast some stuff up there. Here's what some of our filters look like when we filter the seawater at different depths. Here's him working, so you actually get a sense for what we're really actually doing. He's culturing right there. He really likes sunsets, so he went berserk on that. But this was an interesting post, because what he did during the beginning of the summer was Mike went to a summer course in Hawaii. And then he talked about his experience there with all the various people and kind of gave people some insight into what it's like to be there. I know tons of people who have gone to this summer course, and I never know what it was like to be at that summer course, but the blog post really illustrated it nicely, okay? And so what this is doing then for us is it's bringing other people into the world of not just what we're doing, but what life is like on board ships, what it looks like in the Gulf of Mexico, you know, what is going on with all that stuff. So it's a little bit more than just the stuff that we like, but because we like it, the enthusiasm comes out, and there's good pictures and decent editing and that kind of thing. Other things that we do that I think that are useful, I'm going to start here. Joining the lab. Now, I stole this basically from another website, and I credit them up here, compiling funding opportunities. But what we have here is one of the linchpins for any situation in academia, which is money. How do you get the money? How do you get money to do the work that you need to do? We've created a list of as many fellowship opportunities for undergrads, grads, and postdocs as I come into contact with. This is just a great way for people in my lab to find resources, but it's also a great way for the community, anybody that wants to find a list, to come to the site, and it just generates extra traffic, which is useful. I also don't want to have to go look these up all the time, so it's nice to have them in some place where I can use them. The education section, I try to do some different things in my classes. Prokaryote Diversity is Communication Intensive. I put my syllabuses up here as open resources for people that want to do active learning-based kind of processes in their classes. One of the assignments for my students is developing a Wikipedia page on a microorganism that doesn't have one already. These are all the live Wikipedia pages that have been generated in 2 years. Students can link to these on their CVs and say, hey, I went and did that. Here's where it's listed, etc. This is a really valuable resource for people when you're applying to jobs or anything. You can say, look, I created that Wikipedia page. Look, it shows you my scholarship. You can click on one of these and people will go. Okay, let me go back. All right, this must be Colleen's.
You said destructo bacillus, and obviously that's why you want that.
That would be a new one. We'll try to get that. But so what you have here is a nice collection of information. You've got it all linked. You have references. It's all written in the style that Wikipedia would like. And look at all this primary literature referencing that was done as part of the assignment. You know, you go to an employer, or you go to a person who you want to work for in graduate school or something, and you go, look, I've got a writing sample for you. And it's online, and it's still live. It hasn't gotten taken down because I did such a good job. They can see, and I'm serious, they will, on Wikipedia, they will remove things. You know, if you've ever tried to create a Wikipedia page about yourself, it vanishes. I haven't. I know people who have, because you're not allowed to. And there's a large volunteer group of people that go out and police Wikipedia to try to keep it accurate. Definitely try to keep it within the rules of Wikipedia. And so this is the kind of thing where even on our lab webpage, hopefully we can provide some additional resources for people in terms of syllabuses and other things that we're doing. Okay, so that's kind of like a snippet of how we can generate content that's a little bit more interesting for people than just what we're doing in my lab. Okay, got a few more minutes, so I want to share with you one more thing, which is that recently I've also become involved, a collaborator, well, actually we don't actually collaborate, but we are friends and we're colleagues. Jennifer Biddle and I, she's at the University of Delaware, also a microbiologist. created a free online seminar series for microbiologists. And a lot of this was motivated by our desire to see people talk, give talks, interact with people on a sort of a scholarly research base, but without having to go all the way to a different university ourselves or fly them all the way here or go meet at a conference. And so this is the result. We have the other thing is it's all free and it's all freely available and it's all archived and recorded. I'm going to get to that in a second. This is the schedule of upcoming seminars. We're booked out through March. We do about one a month, sometimes we do two. But you will see also that we have previous seminars down here, lots of them. They're all linked to this person's, lab webpage. If they're on Twitter, we've got their Twitter link. And then later on, once they've already given the talk, we have a link to their talk on YouTube. And the way that we do this is with Google Hangouts on Air, which are free. Google Hangouts on Air, it's like a Skype chat, except it's broadcasted live, so you can give someone a link to the live broadcast either on Google or on YouTube. And then the other thing is it's recorded. So you can actually get that thing. You don't have to do any photo editing or video editing if you don't want to. You can if you want to with YouTube's tools, or you can download, you can do anything you like. But the cool thing about it is we now have a list of archived talks. They're freely available, so if you missed one of the seminars and you want to go hear about it, they're all sitting there. People are starting to incorporate these in their classrooms. They're on a wide variety of subjects, everything from rock microbiology to the mouse gut microbiome. We're getting talks about everything under the sun. It's a vast world out there, and this is a really cool integration of a lot of different media. So we've got our live Google Hangout, and some people are actually in the Hangout and talking. We're broadcasting it live on YouTube, but what we also do is we have a Twitter feed, and I want to show you this on my tweet deck, where we created a hashtag called USeminar for Microseminar, okay? And what people can do is they can tweet questions. So we live tweet it while we're going, but other people will actually tweet questions in when they're watching the seminar, and then we can ask the person directly while we're in there, right? So there's only enough room for 15 people in a Google Hangout on air. But what if you have 40 people that are watching the seminar like we sometimes do? Well, what they do is they just tweet us a question to hashtag microseminar, and then I can just ask them because I'm monitoring the feed while they're giving their talk. And so we get these questions in live. While they're on the air, it becomes part of the stream at the end, and it's all recorded. And it's just this really, I don't know, it's very meta to have like the talk on one media and Twitter questions coming in and then slamming them together. And it really feels like you're at a live conference or at a noon seminar or something like this, except you've got people in Europe, you've got people in South America, you've got people on the West Coast, you've got somebody in a room just down the hall from you. Everybody's involved in watching this at the same time. And it seems to be pretty successful and something that a lot of people like. So that is a kind of model that has a lot of room for expansion in my mind. I think that having short interviews with people that were live tweeted or broadcast live that people could interact with rapidly somehow is something that you could do on a variety of subjects. So let's see. Some of you are live tweeting this. Yeah. Perfect. I was hoping that would happen. So there's all kinds of stuff like that that we can do, and as people get more and more international, and it gets harder and harder to bring everybody together in the same place, I think that these kinds of live micro seminar concepts are going to be something that are really valuable to folks. And so it's also a great thing, as an educator, if I want to introduce a topic. It's what better way than to give like a talk on that subject by a world expert on frequently unpublished data that they're just working on now, as opposed to a 5-year-old textbook version or something like that. And so we're slowly creating enough content to really interact with things that way. So I hope you see that we go from like pretty mundane, maybe like focused microbiological specific stuff, but that a lot of these things are really highly relevant to any kind of science topic. And then also, maybe even beyond, you've got a lot of things here that you can do at various stages of your career that would help in terms of making your presence known. Not only that, just expressing yourself. You know, I think that if you can get creative and kill a few birds with one stone about, you know, creating content that's going to be attractive to employers, but also just because you're interested in the topic, right? You want to talk about it with people. Maybe you don't even know anybody that likes to talk about this stuff, but you see some people on Twitter who do talk about it. So you write up some content, you blast it at them, start a conversation, things start rolling. So I think that, you know, personally being at LSU, there's not a lot of other microbiologists here, but I feel like I don't even think about that because there are so many media with which to interact with microbiologists from all around the country now that if I want to talk to somebody in Hawaii or Pennsylvania or wherever, yes, there's email, but there's also a really quick Twitter conversation, or we can talk via conferences like this or things of that nature. So it doesn't feel very isolated when you start putting the feelers out, interacting with all these different media. So I've gone on quite enough. I haven't even gotten any questions from anybody, so I apologize. But we have a couple minutes if you have anything you want to talk about.
Is the TweetDeck an app that you can plug the App Store?
Oh yeah, it's free. There's a few different versions of this. Twitter actually produces this, and you can direct it to hashtags. You can direct it to mentions. You can direct it to specific users. This is my colleague, and we both do micro seminar together. I follow my grad students on there. You know, there's all kinds of things that you can do. You can have your direct messages up there. I have our biology hashtag there, so when people are putting things up that way, you can see that information. So it's pretty useful. Especially if you're doing live, if you want to follow a conference and you don't want to follow all your tweets, you just want to follow one hashtag. Like, you don't get to go to ASM next year, but ASM 2016 will have a hashtag. You can follow the whole conference just with that hashtag. Other things. Okay, well, if you think of something later, you're welcome to get in touch with me. Any of these mechanisms will get you to an email address if you want, or you can always just tweet at me. I'm usually more responsive to that than email, because you can't ignore it as easily.
Well, I'll ask you a question.
It's kind of like being texted, I guess.
So, do you get any media attention, attention from journalists or people who want to cover your research because you're tweeting about it and blogging about it?
I've had a little bit of that, yeah. Yeah, there's definitely journal editors and a lot of science writers follow my Twitter feed. I haven't had a lot of direct, like, hey, let's chat. There's been a couple of them. There's a journalist, a science writer who interviews scientists, and I'm trying to remember the name of the blog right now. Shoot. Basically, it's a 1 hour long oral interview with scientists, hundreds of them up there. It's called People Behind the Science, and they picked me up from the Twitter thing. Yeah, I've gotten asked to review certain manuscripts and things partially because people know about my activity on Twitter. So yeah, it's one of those things where I've gotten a lot of opportunities within the community, but not as many outside of it just yet. But some of that has to do with whether or not we're putting out Nature or Science papers. It seems to be that a lot of science journalists focus on those places and not as much on this. But yeah, one thing to mention, the blogging on there actually got us an award. We got $1,000 in cash for blogging, and so one of the things, and that was just random. Someone said, "Hey, you should send this in. It's a blogging contest." I was like, "A blogging?" All we did is put pictures, and so they liked it a lot. They liked the format. They thought it was useful, and they gave us some money to do research with. And so one thing to do is put yourself into competitions and things, and you'll get more feedback and potentially fellowships and things like that. So that's a good way to extend your blogging reach too.
I didn't even know this existed.
Yeah, there are plenty of contests out there for best science writing, not even science, just creative writing, all kinds of stuff for blogging. Absolutely. Short format, long format, tons.
Well, thank you so much for speaking to us today. I really appreciate it.
Okay.
Thanks.